This microarray hybridization method is useful for comparing a large number of samples from an organism with a known genome. Compared to a high-throughput sequencing approach, the amount of data generated by this method is much easier to handle, and further it can be analyzed in a more cost-effective way. Demonstrating the procedure will be Dr. Christiane Seiler, a postdoctoral scientist working in the research group Heterosis at the IPK in Gatersleben.
Before performing microarray hybridization using the gene expression hybridization kit, prepare 10x blocking agent according to manufacturer's specifications and aliquot the resulting blocking agent in 200 microliter volumes. Then store the blocking agent at -20 degrees Celsius until use. On the day of the microarray hybridization, thaw one 200 microliter aliquot of the 10x blocking agent on ice and pre-warm the hybridization oven to 65 degrees Celsius and a heat block to 60 degrees Celsius. Prepare the fragmentation mix for each sample as described by the manufacturer and mix the samples gently on a vortex. After vortexing, briefly spin down the samples in a microcentrifuge before incubating for exactly 30 minutes in the 60 degrees Celsius heat block. At the end of the incubation, immediately cool each tube on ice for 1 minute before stopping the fragmentation with 25 microliters of 2x gene expression hybridization buffer, high revolutions per minute per tube. Mix with gentle pipetting, taking great care not to introduce any bubbles, and centrifuge the tubes in ambient